Hello, my name is Ashley Duke, and I'm a senior biology and chemistry major here at Radford University. I conducted a metadata analysis of Bacteriocins' effect on bacteria in conjunction with Dr. Joyce Carvin of the Radford University Biology Department. When I first started this research back as a freshman, I began by looking at an article for a class, and I came across the word bacteriocins and immediately wanted to know more of what they were, and if they could be used as an alternative to antibiotics, which ultimately did lead to this research. I conducted a lot of preliminary experiments where I was just learning techniques that I could use when working in the microbiology lab, as well as what had already been done and if I could replicate what had been done when looking at bacteriocins. The purpose of this study, as well as this research as a whole, is because multi-drug resistant bacteria is a problem in treating deadly infections, specifically nosocomial infections in hospitals, and new developments in antimicrobials could alleviate this problem. Bacteriocins have been used to treat produce as well as other areas where bacteria can spread. One study I came across was using a bacteriocin to treat bovine mastitis in cows. But could they be used to treat nosocomial infections and other diseases of human origin is what I specifically wanted to know. So a little bit about bacteriocins. They are of bacterial origin, so a bacteria strain will produce a bacteriocin or some other type of antimicrobial. These can range from bacteriocins to bacteriophages. They have a bacteriostatic or a bactericidal mode of action, which ranges from pore formation in a membrane to nuclease activity in DNA or RNA. And what makes them unique is that they have a relatively narrow killing spectrum compared to antibiotics, which are usually broad spectrum. For the purpose of this study, I conducted an extensive literature review of experiments and review articles. This included compiling a list of about 50 articles that were experiments that had been done with bacteriocins, anything that had to do with an antibiotic and testing it against bacterial strains, as well as review articles about bacteriocins, how they worked, their mode of action, and how they had been used to treat diseases of human origin. Some data that I extracted for bacteriocins, I specifically was looking at their strain of origin since they are produced by bacteria. Their indicator strains, as in what bacteria they have been tested against. Some of those strains included Staphylococcus aureus, E. coli, Listeria monocytogenes, Klebsiella pneumoniae, Enterococcus faecalis, Staphylococcus epidermidis, and Pseudomonas aeruginosa. I was also looking for their inhibitory activity, whether they reported that as a zone of inhibition, a minimum inhibitory concentration, or a percent inhibition. For antibiotics, I extracted data that told me what their minimum inhibitory concentration was, their MICs, as well as what their zone of inhibition was when tested against indicator strains. And I then took the data for both the bacteriocin and antibiotics, converted it into something that was comparable. So, for instance, if I was given an MIC for a bacteriocin, I compared that to the MIC of the antibiotic. If I was given a ZOI for a bacteriocin, I compared that to the ZOI for the antibiotic against that specific indicator strain. I then reported these results in a table. For my results, I did find that about 13 strains of bacteria produced a bacteriocin that was better than the antibiotic that is being used to treat it, or to treat a specific bacterial strain. About 12 bacteria produce a bacteriocin that is worse than the antibiotic in treating a bacterial strain. And what that means is, to classify one as better, the MIC for that bacteriocin had to be lower, or the zone of inhibition had to be higher than what was produced when using the antibiotic. For it to be classified as worse, the MIC had to be higher than the antibiotic MIC, or the zone of inhibition had to be lower than the MIC, or than the zone of inhibition for the antibiotic. I did find 4 strains of bacteria that produced a bacteriocin with activity that was comparable to activity for antibiotics. Comparable, I would mean I classified that as being within 2 millimeters of the zone of inhibition, or where their MICs were relatively the same. There are about 5 strains that I found of bacteria that produced a bacteriocin that needed further research, whether the researchers just did not specifically report their zone of inhibition, their inhibitory activity, or I could not convert their results to something that could be comparable. Some of the antibiotics that I went over in this research were vancomycin, azithromycin, ampicillin, daptomycin, ceftazidimine, rifampicin, and ciprofloxacin. And these were just the antibiotics that I found that were commonly used to treat the specific indicator strains that I was looking at. Some observations that I made: Lactobacillus acidophilus was almost always not as effective as the antibiotic. For instance, against Enterococcus faecalis, it had no inhibition, and that was almost always the case for all strains that I looked at. For Pediococcus pentasaceus, it was either comparable to or better than the antibiotic. So their MICs and ZOIs were the same for one study, and all other studies showed that the MIC was lower for Pediococcus pentasaceus and that the ZOI was higher for that specific bacteriocin than when the same test was done with the antibiotic. Bacillus subtilis was always more effective than antibiotic, meaning its MIC was always lower and its ZOI was always higher than what was produced when tested against the antibiotic. And Bacillus pumilus always showed no inhibition against all indicator strains that I looked at. Some key points that I found in this study: again, some cases of bacteriocin and antibiotic were comparable, whether their ZOIs were within 2 millimeters of each other or their MICs were relatively the same. I did find and notice that there was a possibility of at least one bacteriocin alternative for each bacterial strain that I looked at. I do want to point out that more research is needed for these bacteriocins that do show inhibition, just in order to directly correlate their inhibitory activity with that of the antibiotic. Other things that should be considered is that these results did come from a variety of sources. Not all experiments were the same, not all of them had the same control. Therefore, further controlled experiments should be conducted to directly correlate inhibitory activity of a bacteriocin and antibiotic. And I also converted the results from each experiment for comparison. If I were to conduct further research with this and have a little bit more time to dive into this a little bit more, I would set up a disc diffusion test that I could perform in triplicate to look at antibiotic versus bacteriocin. How I would do that: I would first find the minimum inhibitory concentration of a specific antibiotic that produces the largest ZOI by testing different concentrations of antibiotic. And those concentrations would still be within clinical ranges. I would then determine a minimum inhibitory concentration of a bacteriocin that produces the largest ZOI in order to again test this. I could then set up my disc diffusion test and perform it in triplicate and report the results as whether or not the antibiotic or the bacteriocin produced the largest zone of inhibition for a specific bacterial strain. So thank you guys for listening to my research project. I hope this was helpful, and I hope to do more research in the future.